be the moderator of today's webinar. But before we call Professor Wayan, I want to give a brief introduction about him. So, Professor Dr. Tunas Wayan Artama completed his undergraduate study at Faculty of Veterinary Medicine, Universitas Gadjah Mada, and then he earned doctoral degree in 1989 from Freie University Berlin, Universität Berlin, Germany. And currently, he serves as a professor at Faculty of Veterinary Medicine and also a lecturer at a graduate program, Universitas Gadjah Mada. He also the coordinator of One Health Collaborating Center, leading multidisciplinary collaboration that integrate human, animal, and also environmental health. Throughout his research career, Professor Wayan has received several recognitions, which one of those is from British Council Research Award. His research focuses on the molecular biology of zoonotic diseases, covering a wide range of diseases from leptospirosis and then toxoplasma and also Mycobacterium tuberculosis. He also conducted extensive microbiome and virome analysis in wildlife, molecular studies on dengue virus, and also have a huge contribution to develop the rapid diagnostic test on dengue virus by using NS1 protein. Professor Wayan has authored numerous scientific publications, both published in national and also international journal, that has a contribution to enhancement of One Health research and education in Indonesia. So, wow, very interesting, right? So, without any further ado, please welcome Professor Wayan.
Hello, good morning, good afternoon, and good evening maybe for all of the participants from around the world. There's 10 countries, 11 countries joined for this webinar for today. We have 3 speakers for today. Professor John Peterson from Uppsala, Sweden. Good morning, John.
Good morning.
Welcome from One Health Demonstration Center at Semarang City. And we do hope that you can share your knowledge and your expertise for all of us in the One Health Demonstration Center here in Semarang, and also participants around the world from Zoom. And the second speaker is Professor Wiku Adisasmita from University of Indonesia. And then the third speaker, our colleague, Ibu Parida from BRIN, Indonesia. Welcome, Bu Parida, to One Health Demonstration Center. So on behalf of the One Health Collaboration Center and One Health Demonstration Center in Indonesia, we would like to thank for all participants around the world that join our webinar for today. And in the morning, we have workshop on wildlife and Komodo summer course. But for today, we have webinar. The webinar participate from 11 countries, from Belgium, from Sweden, from Japan, from Cambodia, Vietnam, Indonesia, Malaysia, Pakistan, and Bangladesh. So this is the participants come from many different countries, and altogether registered around 200 participants. And here in the room, many students from UNES from Semarang, and also there's 2 staff from UNES who join us with us. Thank you for your coming, for your presentation for John and Dr. Prof. Wiku, and also Dr. Parida. Before we start this session, I would like to read the curriculum vitae of Dr. John, Professor John Peterson. He is an associate professor at medical virology at the Zoonosis Science Center, Uppsala University. He obtained his PhD from Uppsala University in 2013, where his doctoral research focused on evolutionary origin of flavipiridae, flavivirus, and tick-borne pathogen. His academic journey and scientific work reflect a strong commitment to understanding how zoonotic disease emerged and evolved. In addition to his role in Uppsala University, Dr. Peterson affiliated with the Swedish National Veterinary Institute, SPA, reinforcing his multidisciplinary One Health approach that bridges human and animal health. His research primarily explores the epidemiology, evolution, emergence of viruses in zoonotic potential by applying advanced technologies such as next-generation sequencing and bioinformatics. He has contributed to discovery numerous novel viruses, including coronavirus identified in the wild rodent. Dr. Peterson has published extensively in high-impact journals, including Nature, where his collaborative studies have expanded global understanding of emerging pathogens. His expertise in virology and decision to One Health make him one of the leading voices in predicting and mitigating zoonotic diseases and the intersection of wildlife, environment, and public health. As curriculum vitae, thank you, John, for your participation. Please, time for you to present 40 minutes and then with extended discussion with 10 minutes. Okay, thank you, John.
Thank you. I will try to share my screen. Can you all see my screen? I hope so. So again, thank you. Thanks for the very warm welcome and your invitation to present at your webinar. So today I will have a presentation about a paper we recently published about zoonotic viruses in farmed fur animals, which was done with collaborators in China. And it's actually focused on sick farmed fur animals. And the idea for this actually came quite early when we were making a study on the diversity of viruses that have been published in NCBI GenBank during the last couple of years. So what we did was to actually summarize the number of viruses that are present, if they are zoonotic, potentially zoonotic, or if they are unknown as for zoonotic ability. And we linked them to any farmed animals, the farmed host animal. And what we could basically see then was that for quite a large number of viruses, we consider them that they might be zoonotic. We don't really know. And for some, we don't know at all. So perhaps even half of all viruses that are linked to farm animals, we don't really know if they are zoonotic. So this sort of motivated us to conduct a study on farmed fur animals which have not been looked into. But if we take a step back, how then are zoonosis introduced to human population or animal population for that matter? Well, you can consider that there are perhaps four main modes of introduction. You have live animal markets, where you have exotic live wild animals that might introduce novel pathogens to humans. You have wildlife hunting, which is of course common all over the world, where people hunt various animals for consumption or medicinal products or whatnot. But you also have, of course, domestic animals that are an important source of, for example, influenza viruses. And you also have the part where you have intensive wildlife farming of animals that are actually not domesticated but still farmed. And if we look into live animal markets, well, we all are aware of what happened in China, where while live animal markets were very common, and they had a diverse array of various animals occurring at the same market. So a lot of animal species occurring at the same place in crowded cages, and in contact with each other and in contact with people. So just examples of how it could look like. You had anything from reptiles to hedgehogs to raccoon dogs, and it was actually not that uncommon to have a so-called lunch menu where you could order freshly slaughtered meat from various animals. So just to sort of highlight that often at these markets you have an array of different animal species being present at the very same place, which is a good breeding ground for viruses to be able to jump to different hosts. And as an example of how it looked like in this Huanan market in China, from the very specific marketplace where SARS-CoV-2 is thought to have emerged, they found traces of several different animals, like raccoon dogs, rabbits, hedgehogs, that all showed various degree of positivity. And if you sort of remember this slide with all the animals, and they could further show that the corresponding places where humans actually were found to be infected actually overlapped quite well. So it's just to show that animal markets are really important in the emergence of viruses. But it's of course not only China that has animal markets. Animal markets occur basically all over the world in different scales, and so in some small scale markets you of course have chickens, hens that are being sold, but still you have the dimension of humans coming in contact. And as another example, in a market in Congo, primates are being sold, freshly hunted from the forest. Same thing as an open street market in Congo. You have small reptiles, and you have primates and ungulates and whatnot. So as an overview, we could see that there are also heaps and different types of species being sold, both prepared meat and fresh meat at the very same market. And of course, also in Europe, we have markets. So during the COVID-19 pandemic, we had introduction of SARS-CoV-2 into mink farms, which caused mink farms to cull hundreds and thousands of animals due to fear of SARS-CoV-2 evolving and being reintroduced into human populations. But there are also fox fur production for example, in Finland. Both the red fox and silver fox are still farmed for their fur. But if we look into fur animal production globally, since this is the sort of main topic of the talk, you could see that yes, it's mostly done in the northern hemisphere, in North America, in northern Eastern Europe, and parts of China. And although fur production has decreased quite rapidly, from several hundreds of millions of mink furs in this case to less than 20 million animals being prepared for fur. It's still in the scale of millions, so it's still an important part of how zoonosis might emerge. And if we look into China specifically, you can see that there are perhaps three main animals that they farm: minks, foxes, and raccoon dogs, where perhaps minks are a bit more farmed than any other animal. But just to say that there are many different animals that are being farmed in China, and not all are for fur farming, but it's for medicinal purposes and also for meat. If we look into China specifically, you could see that there are perhaps most fur farming occur in northeastern China, and perhaps in particular in the Shandong province, in Hebei province, and in the other parts of the northeastern provinces. But as you can see, there are also fur farming occurs also in central China and in many other regions. So then again, if we jump back to the sort of how are zoonosis introduced, so our motivation or sort of our reasoning behind this is that yes, we have intensive wildlife farming on the one hand, and we have intensive farming of domestic animals on one hand. But an important link in all of this is that when you have wild animals that may come in contact with either wildlife farming or domestic animal farming. And in our specific study, we looked at the interface of intensive wildlife farming in connection with if they come in contact with other wildlife that are not being farmed. So more specifically, we try to reason that yes, wildlife and farm animals, might be or are probably important hubs for zoonotic virus transmission. But there has been a high focus on conventional production animals like pigs and cattle and other animals. But fur-bearing animals or fur farm animals has not been that much of a focus. And we have therefore not that much knowledge of what viruses that are present in them, although they are considered important for virus transmission. So we saw it as a important need to understand the role of fur farming production animals and how zoonotic viruses might be spread. And this was just a ChatGPT sort of interpretation of how this web of viruses and animals might be visualized. So what we did in our study was to collect samples from sick farm animals. So it's important to remember that in this case, we only had a sample from sick fur farm animals. And they were collected all over China. I will explain later what other and main fur animals mean in this case. But we collected samples from 27 different species and over 450 tissue samples, actually closer to 600 in total. But as you can see, they included many different farm animals. But the ones you can sort of keep in mind are these four main animals: the guinea pig, a weasel, American mink, and raccoon dog. Those are the ones you can sort of keep in mind for now. And when we did our sampling, we tried to sample both intestines and lung mainly. So preferably both lung and intestines from the same animal. In some cases, we only had intestine. In other cases, only lung. But for most animals, we had both lung and intestine. What we did then was to use so-called RNA sequencing. And basically, it's a good tool if you want to capture both DNA-based organisms and RNA-based, and in this case, RNA viruses, because if you consider the sort of that any DNA organism, yes, it has mRNA intermediate, so you are able to capture both the RNA genome and DNA genome by doing this, although it's mainly a tool in our case for viruses. And this RNA-seq has been used for lots and lots of different so-called virome studies. So it was used in the discovery of SARS-CoV-2. We have been using it to characterize tick viromes and mosquito-borne viruses, such in the case of chikungunya. So it's a very versatile tool to use to discover viruses. But in our case, when we sampled different fur farm animals, what did we actually find then? So this is a bit of a messy slide, but if you look into virome studies, this is perhaps the main slide you should look into, or main figure that you should look into, even if it's in the supplementary or if it's a part of the main figures, because it describes basically what was found in which tissue and in which animal. So this is sort of a main result figure where you can see the taxonomy and all related meta information. But I will guide you through what sort of led up to this main figure. So in total, we found 125 mammalian infecting viruses. But before we had this slide, we did first sort of a detailed phylogenetic inference, because that's a main tool where we use it to identify how closely related and how similar a particular virus is in relation to previously published viruses. So we want to know if is it very similar? Yes, then it might be a part of the same species. Is it distantly related? Then it might be considered a new species. So we do detailed phylogenetic inference for all viruses. So in this case, different types of PEGI viruses that was found in mice and in squirrels were considered novel in this case due to that they were not that close to other viruses. So phylogenetics is a really important tool in how to discover novel viruses. But what we could also see that many viruses that we found had a much larger host range than what was previously known. So for example, in the case of the Arctic fox, which is, let's see where it is, sort of in the middle, it was found to carry 13 different viruses in total. And the same for the guinea pig. It was associated with 18 different viruses. So a sort of a key message in this case was from our study was that although you might perceive that some viruses are only associated with a certain host, what you can see is that many viruses actually have a broader host range than what you actually think. So that's a good thing to keep in mind, because what we could see also was that co-infections are quite common. So although for most we could only find a single virus, but for over half, two or more viruses were found in each so-called sequence library. And this varied a bit between species, where the sort of main suspect, the guinea pig, mink, and raccoon dog, often had quite a few number of viruses. But we could also see that rabbits, this Cuniculus, also had quite a few, were often infected at least with a single virus, but often with two or more. So it's important, yeah, different animal have a different infection load, so to say. Not all animals have the same amount of viruses in them, but many have more than you expect. Another thing is that you could clearly see that different viral families have different virus tropism. So if you consider the sort of turquoise color, you can see that coronaviruses, yes, you find them if you sample lung tissues and intestinal tissues. But if you are interested in paramyxoviruses, then you should definitely sample lung tissues over intestinal tissues. And conversely, if you're interested in pseudoreoviride viruses, yeah, then it's a good thing to sample intestinal tissues over lung tissues. So that's also an important thing to keep in mind, that different viruses have different tropism. So depending on what you are after, if you are interested in the broad scale picture, or if you are really targeting something, then you have to know what tissues to sample. Another thing we tried to do was to identify so-called risk viruses. So one thing was that we wanted to characterize known zoonotic viruses, and there were quite a few different kinds of alpha coronavirus 1, beta coronavirus 1, Japanese encephalitis, rotavirus, and they were found in different tissues and in different animal orders. But then we also wanted to sort of more focus on risk viruses. And in this case, we categorized some viruses cross-order viruses. Those were considered viruses that they are not found in humans, but they are observed in 2 or more animal orders, sort of as an indication that they are prone to host jumping or might jump into different hosts. And we took this a step further and had so-called high-risk novel viruses. So they have not been described prior to this study, but they were found in 3 or more animal orders. So a clear indication that they might be able to jump into different types of hosts. And there were in total 13 of these viruses that were found in different hosts. They were primarily found in lung tissues and in intestines, and primarily in rodentia. So if we take it again a step back, we could see that many viruses or many viruses in this case, had a particular broad host range. So for example, rotavirus A was found in a huge number of animals, many more than expected. And same for these Amdopa viruses. We found them in several different mammals. So it's just good to keep in mind that, again, many viruses have a broad host range, and many hosts have the ability to be sort of carriers of many different viruses. So if we look sort of specifically into the raccoon dog, we could see that it carried, I think it was 18 different viruses, and was often commonly infected with alpha coronavirus 1 and this amdoparvovirus carnivorum virus 3. But again, you can see that the raccoon dog itself had many different viruses that it became associated with. Like this Jeta virus was a novel virus not previously found in raccoon dogs. And same if we looked into mink, it also had 20 different viruses associated with it. And in this case, in mink, we also identified a quite interesting virus, this Pipistrellus bat coronavirus HKU5. And why is this one interesting? Well, if you sort of have the SARS-CoV-2 and COVID-19 in mind, this virus had only been found previously in Pipistrellus bats, so no other mammalian species had this virus before. And in our case, it was collected from mink with severe lung disease. So already associated with disease. So we speculate that it might have been similar to SARS-CoV-2, a potential transmission of this virus from bats to minks. So a bit of a warning sign in this case. But we also found many other coronaviruses. So in total, we found 7 coronaviruses in 66 different animals. So this mink coronavirus 1 was found in 18 different mink, and canine respiratory coronavirus was also found in raccoon dogs. So coronaviruses are found in many different animals. We also found influenza A viruses, H5 and N6, and H1N2 in various animals. So in minks, for example, we found H5N6 in 2 minks. And in this case, the phylogenetic inference indicated that this might have been due to reverse transmission of contact with animals sorry, it might have been avian derived originally, as it was indicated by phylogenetic analysis in both the HA and NA segment. And the phylogenetics analysis of the HA segment indicates that it belonged to a subclade that has been spreading globally and caused quite a bit of concern. And in the case of guinea pigs, sorry, I went too fast. In the case of guinea pigs, we found H1N2 in guinea pigs. And this was quite an interesting case because in this case, the H1 fragment actually clustered with H1N1 from 2009, while the other gene segments clustered with swine viruses. So in this case, it was suggested that this type of reassortment could have occurred in swine following a reverse zoonosis event. So actually from humans to swine and then back to guinea pigs. And another warning sign was when we looked at the phylogenetic tree of the HA gene was that it might have actually been circulating quite for some time or an extended period of time. And all of these animals, as we mentioned, had respiratory symptoms. So again, an increased risk of disease transmission to humans. That's a peculiar finding. We also found Japanese encephalitis virus in guinea pigs. It was a completely odd finding, and we don't really know how it ended up in a fur farm guinea pig. But that's just to say that these viruses are really ubiquitous and can be transmitted to different hosts or hosts that we don't really expect to become infected with these viruses. So again, we consider this interface basically of wildlife being in contact with intensive wildlife farming or with the domestic animal farming to be a really, really important hub in how zoonoses are introduced to human populations. And that's not to say that live animal markets or wildlife hunting is important. It's definitely important. But wildlife that are able to introduce viruses to animal farms or to domestic animal farms have the capability of really, really causing viruses to adapt to human population and thereby cause the emergence of novel zoonotic diseases. So I've been talking perhaps a bit too fast, but just to summarize, we found 125 viruses in our study, and 36 of them were novel, and we considered almost 40 of them to be at the risk of high risk of spillover between species. But we also indicated that some animals are at higher risk of being sort of reservoirs for zoonotic viruses than other. And we also could clearly see that some specific viral families, Coronaviridae and Paramyxoviridae and Pseudoreoviridae, are able to infect many more hosts than other viral families are. So if you want to read more about the details of this study, you can have a look into this paper or contact me. And with this, I want to thank you all for listening, and I thank everyone who has been part of the study also, and the funders that have helped to produce this research. And with this, I open for any questions, and back to the organizers. I will stop sharing also. Thank you.
Okay, thank you, John, for your presentation. We
Thank you.
would like to invite the participant to ask question to John. Please, who will ask question for him? Maybe before waiting for the question, I want to ask some question to regarding the virus that now we do have trend viruses in the Congo, Cameroon Congo hemorrhagic fever virus. Is that transmitted through migration bird and also through ticks? Is that widespread spread to the Europe? According to the news, is already spread to the Europe. How about you comment, John, regarding this outbreak in Congo?
Yes, it's interesting to see how many viruses actually have become established, perhaps in particular in Southern Europe. So there's been continuous outbreaks of Congo Crimean hemorrhagic fever virus. West Nile virus is now very present in south of France, in Greece. Dengue virus has become almost endemic in many regions. So you can see that the borders for many viruses are not as strict as they were before. For only a couple of just a decade or 2 decades ago, we would not expect to have viruses occurring in Europe, but we can see probably as an influence, as a consequence, due to climate change, many viruses are pushing their borders, and we will undoubtedly see cases occurring even further up north in Europe.
In terms of vaccine for like COVID-19, we do have a vaccine for COVID-19, but for these viruses, we don't have vaccine yet. And then how the preparing for that to minimize the transmission from the reservoir or the vector to the human being?
Yeah, exactly. So while we are waiting for good, affordable, and effective vaccines, I would say that surveillance is what we need to focus on. So we need to know what's out as for total diversity, what viruses are circulating among our animal population and in even our vectors, our domestic animals, our farmed animals, our wildlife. We need to know what's out there to be able to prepare for it. But we also need to conduct regular surveillance of known viruses that occur in known vectors or known reservoir animals. So it is a it is a huge and costly effort to do so, but if we want to stay prepared or if we want to prepare for the next pandemic, we need to put focus on surveillance and exploratory studies.
Yes, yes. The key must be we should have surveillance, molecular surveillance for that viruses. Okay, thank you, John.
Thank
Any
you.
other question from the floor here? Please.
Okay. Is it honorable to ask you, Professor John? My name is Azmiula Nirwastasari and I'm from University State of Semarang. Okay, I will ask about the zoonosis. Basically, in this century, as we know, several cases of zoonosis have been reported. I will ask, what's your opinion and what the role do the veterinarians and wildlife biologists to early detection of zoonosis outbreaks? Thank you.
Yes, thank you for the question. So I think they play a very important role in being sort of the, what do you say, the first responders, basically, to be able to see if do they notice animals that are sick or have symptoms that might indicate that they actually are sick. So they are sort of our first responders, basically, to see if something within our wildlife or domestic animals are uh, have some type of zoonotic disease or might potentially have a zoonotic disease. So they are very important in the overall sampling and surveillance systems, I would say.
Okay. Thank you, Professor John.
Thank you.
Okay, any other question from the floor from here? Okay, Sabrina.
So, hello, John. That's a very interesting and extensive research that you have done. I have a questions because from your research, you presented that most of the virus that have the zoonotic potential will be the RNA virus, right, such as the corona or Parmixo. Well, I would like to ask you, how do you think is it essential for DNA virus to play a role in the zoonotic potential? Because when I did my PhD research, I did a study about the domestic cat hepatovirus, which is the same family as human hepatitis B virus. And one of the surprising findings that we have is that they shared a conserved viral entry receptor. So, and then when I try to isolate the virus in the human cells, the human hepatocyte cells, surprisingly, the virus can infect the cells. However, the subsequent replication is limited. So I just wonder, how is your opinion about the risk from the virus to be a zoonotic potential nowadays? Okay, thank you.
Thank you. I definitely don't think we should neglect DNA viruses in all of this, in looking for novel zoonotic viruses. So DNA viruses are undoubtedly important. I think the main difference is that RNA viruses have perhaps a slightly higher ability to evolve due to the difference between the RNA-dependent RNA polymerase and the DNA-dependent DNA polymerase, that you have a better error correction in the DNA viruses than you have in RNA viruses, which allows them to accumulate novel variation, and therefore they are able to perhaps better adapt or quicker adapt and have higher diversity. At the same time, you have many undiscovered DNA viruses. But to me, whenever we do these studies now, we always try to focus on looking into both DNA virus diversity and RNA virus diversity, just because we shouldn't exclude DNA viruses from these analyses. So I agree, they are very important.
Can I have a follow-up question?
Yes, thank you.
So another question is that DNA virus such as hepatovirus, they say to have like cell tropism and host tropism, which some of the researchers says that human hepatitis B virus can only infect humans. Even in primates, it's very difficult to infect the primates. However, when we study about the domestic hepatovirus, they show that they share a common viral entry receptor. Do you think in the future, the domesticated hepatovirus might also have the zoonotic potential to the human? Thank you.
It's a bit outside my field, but it's not impossible. That's for sure. Given enough evolutionary time, so to say. But yeah, I will stop there and say that it's perhaps not unlikely, but we'll have to wait and see.
Thank you so much.
Okay, thank you for your question. Anyone from the Zoom want to ask to John? Maybe Dr. Rita. Dr. Rita is one of our group in the parasitology network. Maybe Dr. Rita or Prof. Wiku want to ask something to John?
Yeah, my chat, Prof. Just I wrote it in my chat.
Prof. Wiku, the next speaker, you want to ask something for John?
Thank you. I'm just listening. Thank you.
Okay. Anyone want to ask question to John, because John is far away from us here. Is from Sweden. Okay. If no other question, I would like to thank to John for your presentation. Very nice presentation, and we will continue our collaboration for the next step. And then I will thank you again, and
Thank
I will
you.
next speaker, Prof. Wiku, for the next speaker. Before Prof. Wiku, thank you again, John. So for the next speaker, we have Prof. Wiku Bhakti Adisasmito, MAC, PhD, Professor of Health Policy and the Faculty of Public Health, University of Indonesia. And he was our coordinator of One Health, Indonesian One Health Network. We call that Indohun. He carried his doctoral of veterinary medicine from IPB University in Bogor and obtained master and PhD at the environmental health from Colorado State University, USA. With extensive expertise in health policy, system management, and zoonotic disease control, he has published numerous scientific papers and actively promotes the development of One Health education and research in Indonesia. Prof. Wiku also served as a young professor at the Tufts University, USA, and affiliate professor at the University of Minnesota, USA. He plays a key role during the Indonesian COVID-19 pandemic, respond as the head of the expert team and spokesperson for the National Task Force of COVID-19. At the international level, he is a member of the One Health high-level expert panel. We call that OHCC, established by WHO, FAO, OA, and UNEP, reflecting his global leadership and health governance, pandemic preparedness, and One Health implementation that integrate human, animal, and environmental health. Okay, time for you, Prof. Iku. Thank you for your coming.
Okay, thank you very much, Prof. Wayan Tunas Artama, for the introductions. Good afternoon, selamat siang for the audience. I apologize that I have to join through the Zoom. I was going to come to Semarang, but due to several meetings here in Jakarta, so I couldn't come to Semarang. So I'll deliver the presentations through the Zoom. Prof. Ayan, should I speak in Bahasa Indonesia or in English? My presentations is in English, but I can speak in Bahasa Indonesia if you prefer for me to speak in Bahasa Indonesia.
Okay, maybe you can mix Pak Wiku, Prof. Iku, because there are participants from 11 countries around the world. Maybe better speak in English.
Okay, thank you. Okay, so okay, I will speak in English. Let me try to deliver the materials with the title of Control of Zoonotic Disease based on One Health approach. But my presentations will not be very technical on biological aspects, but I will be presenting more on the organization's matters related to global, national, and local in Indonesia. I hope that this content of my presentations enlighten you all from either from Indonesia, especially, but also for others' perspective for the international forum. So the issue that I'd like to present to you basically is that first, I'm sure you all understand that there is a linkage between the environmental health and health issues. That the climate crisis, because of the deforestations, because of the poor waste management and utilizations of the fossil fuel-based energy, can cause disease agent to be much more easier to infect. And then the risk groups are more vulnerable, and the weather and environmental anomalies arise, and we are experiencing right now in many parts of the world, including in Indonesia. Of course, it will disrupt the animal habitat, and then the new agent transmissions from the animal to humans are arising, and then the risk groups also are more vulnerable, especially the comorbid society, the poor society with very low hygiene and poor environment. And then, of course, the problems would be more zoonotic diseases transmitting from environment, animal to the humans as at the end. Although that's also the vice versa, it will also cause the other way around. So the emerging infectious disease is now, because of the COVID-19, we know it's a global threat. And that's why the international countries right now, through the World Health Organizations, are discussing on the how are we going to manage our world to prevent to have the pandemic in the future. So I will explain that later, but at least because of this course of the diseases over time, there are many diseases that we are facing of the emerging infectious disease, and more because of the climate change. Of course, in Indonesia, for example, in the peatland in Sumatra, especially in Kalimantan, those are the reserve of the microorganisms with a different environment. So when the climate change occur, then it may change the situations so that the bacteria, viruses, and other microorganisms are being exposed to the different environment. Of course, it will be hard for them to survive, but those who are survived, which fit with the environment, if they want to survive, then probably they can disrupt the human activities. And for that, that's why One Health collaborations is important. And the One Health collaborations, not only at the global level, which now has been empowered through the collaborations between FAO, World Organizations for Animal Health, UNEP, and also World Health Organizations with the One Health High Level Expert Panel, or OLEP, but also this has to be strengthened at the regional, national, and local level. Imagine that there are hundreds of countries in the world. It doesn't mean that all countries or national systems and governance are 100% in line with what the global commitments at the UN levels. So that's why what is important is not only at the global level, but more importantly at the local level. So how can we organize orchestrating the actions for One Health from central to local, or from global to local, or from local to global? So vice versa. We need to make sure that there is a connections between global governance and the local governance, because otherwise, with the experience of COVID-19, everybody observed that if one country can solve or better solve the problems of COVID-19, that doesn't mean that the region and the global can deal with that securely, like one or two countries at the beginning they can handle the COVID, or vice versa. If we can see that there are many countries, probably they cannot handle it because there was no collaborations, strong collaborations assisting each other across countries. So imagine in the world in the future, with the learning of that, it should be improved. Same thing when we are talking about ASEAN, the region. The region has been committing to have a collaborations on One Health. However, at the regional level, that doesn't mean at the local level has the same concern, connections, preparedness in terms of responding to the outbreaks or the emerging infectious disease, zoonotic disease, etc. So that's why the collaborations, the multilateralism across countries are very important in terms of the policy, in terms of preventing the encroachment or interventions to the forest environment, for example, to the marine, because when the habitat is damaged, then the crossing of the microorganisms will occur as well. That doesn't mean that it's 100%, but it may change a lot when they can survive. They can survive with the severe environment or conditions. So that's why the quadripartite, WOA, FAO, WHO, and also UNEP developed the One Health theory of change. So this is the theory of change, of changing infrastructure, ecosystems at all level, from the organization's level up to what is happening in the field. Why we have the One Health theory of change? Because we know that the connections between sectors are very important, and the world has to be better to prevent, to predict, to detect, and respond to health threats. So, for example, like the potential pandemic in the future, we have to make sure that we are dealing with the environment that not necessarily can be dealt by only one sector. Let me give one example. When we are dealing with the biodiversity issue is regulated through the at the global level through the Convention on Biodiversity. Convention on Biodiversity is organized or managed by the United Nations Environmental Programs, where the representations of each country are coming from the ministry in the field of environment and forestry. It depends on the governance of each country, but mostly they are from environment or forestry or environment and forestry sectors, where they have the surveillance system by themselves. Whereas when we are dealing with the zoonotic disease, then the surveillance from the animal sectors, including from the sector of the agriculture, for example, livestock, those has to be shared with the surveillance systems and governance in the public health sectors or human health sectors. In many places in the world, including Indonesia as well, the connections between at least 2 to 3 sectors are not the same connections at the global level. So that's why improvement of the multi-sectoral connections and collaborations are very important, because the global level are harvesting the result of good results of the local connections or local coordinations. So this is the theory of change, to make sure that we understand the changing of the ecosystems, the equilibrium. It will cost something. So that's why we need to adapt with this. For this, we need to prepare. We need to have early warning systems, the notification systems, the response system, and the recovery. This is not only for the human health sectors, but of course, for the animal health sectors as well, environmental sectors as well. All together, human, animal, environmental sectors, we need to improve the capacity, not only from the, if you ask, from the animal health sides, but also on the human health side and the environmental sides equally, to be able to prevent, detect, and respond. If we have the ability in all area to prevent, detect, and response, and of course, this will improve the resiliency of the public health if we see it that the human as the final, cause of the damage later. So the World Health Organization has been issuing a treaty. They call it a pandemic agreement or pandemic accord, or pandemic preparedness and response of the potential for the future pandemics. So there are 35 articles, 3 chapters, and the chapters, the articles that I highlighted green, Article 4, Article 11, 12, 13, and also Chapter 3, those are the chapters and articles that are very contentious. The issue is related to the pathogen access and benefit sharings, the issue of the intellectual property rights, who own the intellectual properties if they are coming from the biodiversity, whether it's if it's country origin and it's found by researchers from the one university, for example, then is that owned by the university? Is that owned by that area? Is that owned by that country? Or it's owned by the global? Because when we found that microorganisms or virus, for example, then if we develop the vaccines, then who owns the vaccines? Who owns the microorganisms? Who developed the R&D? And then who manufacture it? And then who is going to buy it? And where the funds comes from if there's a pandemic. So those are the things that are discussed. And now the World Health Organization is discussing on the annex, because the pandemic agreement has been agreed by all member countries, 194 countries. But the issue that has been discussed starting from 2 months ago until next year on the World Health Assembly in May, that they have to agree on the annex of the pathogen access and benefit sharing. Now I'm going to discuss about the pathogen access and benefit sharing. These two issues, pathogen access and benefit sharings, several countries several big countries would like to entangle these two. So it's a separate one. Pathogen access is different with the benefit sharings. But many group of countries, countries from Africa and Group for Equity, including Indonesia, would like to have pathogen access and benefit sharing as one issue, cannot be separated. Because if we do the surveillance and find the pathogens, potential pandemic pathogens, which is sometimes we don't know whether that's a potential pathogens for the pandemic or not. But if we find it because of our surveillance system, for example, like Indonesia or Congo or any countries in the world, then if it's developed into vaccines in developed countries, not in the developing countries, because there is a collaborations probably with the advanced, mature, good universities from the developed countries, from the US, from the Europe, for example, from the Western world, then and then manufactured over there or somewhere else, then who will pay if we want to get the access of the vaccines? Imagine big countries, not necessarily they have sufficient funds to purchase the vaccines. So the one who can develop the vaccines, those can sell the vaccines. But if the countries or the people who are not from the so-called rich country or developed countries, then they will be limiting access to the vaccines and drugs. So that's why the ownerships of the pathogens found is a contentious issues, because developing countries, so the pathogens usually will be found in the country where biodiversity rich, like Brazil, like Indonesia. At least those two countries and many other countries in the world are biodiversity rich. So potential pathogens are found over there. But the ability to have good surveillance system is limited. They need also to have funds. Where they get the funds if they don't have the funds? So they need to get the funds of selling the drugs from developing from the pathogens. So that's why the mechanisms of sharing is key. You know, when we are dealing with the infectious disease infecting humans, then probably everybody will say, well, there is no commercial behind it. Always there is a commercial behind it. We need to access it. We need to make sure that the world agree of a certain mechanisms so that people can access. So these are the issues of the intellectual properties, the transfer of technology, know-how. It's always associated with the cost, with the funds, and the procurement and distributions, because not during the pandemic, the distributions will be halted because of the transportations disruptions, etc. So the supply chain, the logistics are problematic. Those are the issues that have been discussed, but the main thrust or the main issue of the pandemic agreement is basically the PAPS, pathogen access and benefit sharing. So the surveillance of the animal agriculture in connections with the ability of the R&D and productions. And then later is the procurement and distributions. So that's, and this is the mechanisms. So imagine the mechanisms that being discussed for the PAPS. If you are familiar with accessing on the film, the theater, the film, the movie. You can access Netflix or others, Spotify for the songs. Imagine that everyone who enter their email address and the payment systems, they can pay according to the time of the access, the value of the movie itself. And then the funds will be transferred appropriately considering who developed the movies. So that kind of platforms, imagine that's in the music industry, in the film industry. Now we are in the PAPS industry or the pandemic potential pathogens industry or vaccines industry. And the mechanisms we can learn from there, because it has to have some trust. It has to have a mechanisms to share the benefits. It has to have a mechanisms of identifying who are the first creator or who is the first finding the pathogens. So this is the mechanisms, laboratory, universities, the government, etc. But then, what can be shared? They can be shared with the GSD, genomic sequence data. So, these are the terms that has been used in the Convention on Biodiversity, now being adopted in the World Health Organizations. Imagine that's under the UNEP systems. And then now World Health Organizations are developing that. Because if the terms are not harmonized, how can then each country and governance at the local level, they can be followed? Because, for example, in Indonesia, we have a local office for forestry, for example. The name of that dinas might be Dinas Kehutanan or Dinas Perkebunan or Dinas Lingkungan Hidup, which is probably they don't know about genomic sequence data or digital sequence information that regulated under the Convention on Biodiversity. Similarly, the office responsible for public health, Dinas Kesehatan, for example, in Indonesia, they don't deal with the GSD and DSI. But when it comes into zoonotic and emerging infectious disease, they need to know. Because otherwise, if they share without notice, then later becoming a product, we need to be able to trace back who's the first access or sharing that informations. Because sharing the pathogens, not necessarily with the biological or the physical sharing, because using only the genomic sequence data or digital sequence information, we can share it through email, we can share it through WhatsApp, and other mechanisms. But when it's registered, like what is in Netflix and Spotify, then it is identified where it has come from. So that's more or less the mechanisms. Now I go to the Indonesia context, and I'm sure in other countries it's the same. What about the quarantine systems? In Indonesia, we have Badan Karantina. And Badan Karantina or Indonesian quarantine authority, they have animal, plants, and fisheries. And there are lists of pathogens under that. Imagine that in the border, whether it's in the seaport, airport, or cross borders, they have to have the ability to screen of those potential emerging infectious diseases. These are the list. Not necessarily they are zoonotic, but if they are zoonotic, are you sure that each border has the ability to screen? If it's not, if for the Indonesians, if you read the news lately, there was contaminations of the shrimp that being exported to US and with contaminations of cesiums. And then when it goes down to where it comes from, traced back, then it was coming from an area in West Java. So those are the things that's becoming an international issues because the ability to screen is late. So imagine if this is a pandemic situations when the pandemic potential pathogens is crossing from one country to the other without the ability to screen it. Because if you are talking about surveillance, what kind of surveillance? How can you screen that when you have the many list? You have limited, people. So that's why you need to know, you need to employ the technology. What kind of technology? Combinations of digital and genomic, because you need to know the genomic sequence of important potential pandemic, pathogens that are, pandemic potentials. You need to know the sequence, and then the sequence has to be tested or matched with the commodity or anything, goods that are crossing, including information. How can you screen the informations from the WhatsApp messages that are crossing across countries within seconds? So those are the challenges now and the future. So that's our task together. So these are the list, easy to say, select agents, these are the list, but crossing to the borders of many countries in the world, we need to have a global surveillance system. When we are saying global surveillance systems, then we are talking about national sovereignty, kedaulatan sebuah negara. Are we going to share that? If those organisms, microorganisms, have economic values for that. So that's why we need to have collaborations, we have to have the prevention systems, the preparedness, and response systems. So that's more or less that I'd like to convey the message that the surveillance points, institutions, officers in all level, like in Indonesia, need to make sure that they are strong enough and have the technology to screen, whether that's related to animal, plants, aquatic, humans. Human-animal environment interface in actions is for the commodities that are crossing. So this is the details, but the end, that what I'd like to enlighten in this presentation is that we need to have multi-sectoral traceability system to improve the biosecurity of each region. I'm not saying only country. If we are talking about Indonesia with the archipelago, we need to improve the biosecurity of each area, whether it's an island, groups of islands, or a country, or region. Because if we secure the biosecurity of each areas, then we secure the world. So that's why having the collaborations between, quarantine divisions, agriculture, human, forest environment, all together into one data, so that we need to have the digital collaborations as well, so that the innovations between genomic and digital all together into one data. So that's the learning from COVID-19. So thank you very much for your attentions. I hope you enjoy with these presentations, and I'm ready for the questions. Thank you. I give it back to Prof. Mayan.
Okay, thank you very much, Prof. Wiku. The very nice insight of regulation of the zoonotic diseases and emerging infectious diseases. So the time for discuss with us. Before asking from the Zoom, the participant from the Zoom, I would like to ask first question to Prof. Wiku. Prof. Wiku, do we already ready with preparedness and prevention and also respond for the incoming emerging infectious disease through our regulation and in the national, international networking, because we still have a problem in One Health in the government level. Like what we have human resource for One Health, they have already trained One Health, but after promotion they have another job and they forgot their job for the maintaining of a response and also prevent the emerging infectious diseases. What is your opinion according to experience, a lot of experience in the managing of COVID-19? And you are very responsible at that time, introducing how to manage the COVID-19 at that time. Thank you, Pak Wiku.
Okay, thank you, Prof. Mayan. If you ask me whether we are ready or not, in a positive way, I would say it's much better than before. Whether we are fully prepared in an idealistic way, of course not. But we are much better than before because we have experience of facing COVID-19, of collaborating across sectors from a very sectoral approach into multi-sectoral approach. When we used to have only 4 diagnostic laboratories under the Ministry of Health detecting for COVID-19, up to at the end after 3 years of handling COVID-19 through task force, national task force of COVID-19, we ended up with 1,200 laboratories connected across Indonesia. So if you ask, are we well prepared after the COVID-19? Of course, 4 compared to 1,000, more than 1,000 laboratories. Of course, the potential, the improved capacity of detections improve a lot. The capacity to response, of course, it's improved a lot. Response in terms of human, catastrophic, situations with a lot of casualties, people sick, etc. Prepared, while this is not like an earthquake that's only occur on only several minutes. COVID-19 are in months and years, so that's why detecting and also preventing along with response all together. It's not only in Indonesia, but it's all countries in the world. And the progress, the learning process, of course, is different. Indonesia, in my opinion, progress very fast and very far. And we are big countries, but because of the collaborations across sectors, and COVID was not like what we are thinking that, well, probably this is zoonotic, that the animals is there and the human interactions is there in Indonesia, for example, then it's very obvious. It was not like that. But if that is like that, then probably your questions would be very fit with the situations, whether the officials from the Ministry of Agriculture, for example, with Ministry of Health and probably Forestry, all together working together, and also with the Ministry of Transportations because of the people mobility restrictions during the pandemic. So that's my response, Prof. Mayan. Thank you.
Okay, thank you, Prof. Wiku. Any other question here from the floor? Bayu, you want to ask question, please? Bayu is one of our staff at the OHCC UGM. Please.
Okay.
Prof. Wiku, first of all, I want to say thank you for the interesting presentation. So my name is Bayu, one of staff, One Health Coordinating Center. So my question, as long as I know that One Health need a lot of resources and also funding, for example, to conduct a regular surveillance, monitoring, perhaps also molecular detection of pathogen, etc. And it is just my curiosity as a person who has a lot or has a bunch of experience working with the government, Prof. Wiku. what is the bottleneck in establishing One Health approach in government level, and also how far the government commitment toward this issue? Perhaps we can take into account the in terms of investment of research, for example, or the screening process, the surveillance system conducted in Indonesia, etc. Thank you.
Okay, thank you, Bayu, for the questions. So, One Health approach is not cheap. It's easy to say One Health approach. In reality, in operation, it will be costing a lot because of the laboratory capacity, the capacity building of the people. You need to train a lot of people, not only from one sectors, but multi-sectors. You cannot only imagine human health, animal health, and environmental sectors, including from the transportations, including from the borders, from the army, from the police. So basically, it's all because the threat is among us. It's in our ecosystems. When we have to control ourselves. So that's why it costs a lot. But if you are asking about the government commitment, they commit very high. However, whether the policy or the programs financing are supporting or not, they are supporting, but still limited. So I met with the coordinating minister of human welfare, Kemenko PMK, yesterday. I met him, and he asked me to present on the One Health approach in front of him and his staff to make sure that the approach of One Health fit with the national challenge right now, which is with the tuberculosis. Imagine tuberculosis, the number of the people die because of infected by tuberculosis in 2024 is 125,000 people die. And the question is that whether the approach of the Satgas, the task force of COVID-19, of the one command, one narrations, one data can handle the tuberculosis. So that is the question. So basically it's the One Health approach with the task force approach in handling COVID-19 being questioned whether that approach would be a good approach for the future. And let's see what's going to happen in the future. So I hope that I answer your questions. Thank you.
Okay,
Prof.
thank you.
Wayan?
Here in the Zoom already, Dr. Asik raising hand. Maybe you
Okay.
can directly question to Prof. Wiku. Dr. Asik, please,
Yeah.
your time.
Yeah. Thank you. I just want to open to my display. Thank you, Professor Wiku, for your really important and nice presentation for us. But learn from COVID-19, I think it's really important for us. Currently, I think after COVID-19, we still have kind of the outbreak of the disease, like the rabies or dengue. I think it's every year we are facing for this outbreak, and I think learn from the COVID-19. I think it's really important, as you mentioned, that it's really have how we are integrated from the data and intervention. I think, and also it's really important for us is the team. When we are, as our experience so far, that when we come into the how handling the outbreak, especially in the local government, we see that the mission, the value of the COVID-19 that we are should be one integrated, holistic, intervention is not really. It's not really happen today. I mean, they come into the silo silo approach currently, and then a lot of them is just handled by the health sector, meaning here is Dinas Kesehatan, I just only see one a district at that time that really good also in the team. Sintang Kabupaten, I think at the time when we are trying to exploring for the dengue outbreak at the time. So the team is led by the Badan Pencegahan Daerah ya, Badan Pencegahan Bencana Daerah. So they are really good one, but not happen in the others the district. I think this is I think I really agree with your opinion, with your idea and so forth. But how we can starting from this issue, I think it's really important to make it like the one paradigm so far and come into the not normal but new normal that we are in different level of the preparedness. Thank you, Prof. Iku.
Okay, thank you, Dr. Asik. Yes, probably you have a lot of expectations as public would like to have that collaborations remain after the COVID-19. However, at that time, because of the COVID-19 with the task force at the central level, national level, as well as the local level, the orchestrations happened during the COVID-19. When that over, then the systems or the challenge are not there anymore. So that's why they are going back to the previous mechanisms. So that's why training, retraining, exercising, and get ready for anything happen to the future need to be enforced from the central governments to the local governments. So that's what I can say. But with the local disaster agency BPPD, which is the part of the BNPB, of course, since their main task is for the disaster response, of course, their approach is very, very collaborative. So whether we want to improve that, because there are a lot of potential disasters in Indonesia, landslide, earthquake, flooding, including communicable diseases. So that if that is the policy from the central government to be employed in all local governments, of course, the collaborations across offices in the local governments will be improved as well. But that's the policy from the central. If they feel that this approach is important to be part of the governance systems in Indonesia. Thank you.
Okay.
Thank you, Professor Iku. Thank you, Professor Wayang.
Yes. Okay, any other question, Dr. Asik? No? Okay, here from the floor, Bu Farida want to ask question. Please.
Thank you very much, Prof. Wayan. Good afternoon, Prof. Wiku. Thank you very much for your presentation. I'm Farida from BRIN, Prof. Previously, I was a researcher from Balitbangkes, Prof. So kind of like a little bit difficult to communicate to how can I say, present our research results actually, Prof, to the Ministry of Health right now. So do you have any idea how we are now in BRIN can share our research results to the Ministry of Health in terms of One Health of zoonosis? Thank you very much.
Thank you, Bu Farida. My quick response to you, and very serious, but it can be also a joke. Just call Pak Budi Gunadi Sadikin. Of course, he will listen to you. As long as your research result is very, important, related to the potential emergency to come. I'm sure if the findings is very key, I'm sure he will listen. I mean, the institutions will listen to you, especially the minister. One of the examples, if your research finding, as I, what you call it, proposed, if you can have an innovation of the digital genomic systems that can identify the potential pathogens crossing by very quick testing, I'm sure that will be a good innovations, because the BGSI, used to be Ekman, basically is a genomic laboratories. So, but they don't have the digital systems yet, like what we have in GSAID. For example, in GSAID for the influenza and also COVID-19, they have the digital informations or sequence. So when they test using the PCR and match, they can give you exactly where it comes, where it was first found and reported by whom. So imagine if you have the list of the pathogens, as I mentioned, under the Badan Karantina, on plants, animal, and also fish, then it will help a lot in screening in the borders with the digital, so that we don't need to have many labs, many people are, in the border, but the system itself digital and can be accessed everywhere in Indonesia, and secured only for the collections of the Indonesian microorganisms important under watch. So if that is your innovations, I'm sure everyone will listen to you. So that's one of the examples. So it doesn't have to be, don't give something that it's already, we know, everybody knows. Make something different. Thank you, Bu Farida.
Okay, Prof. Wiku, the last question here from the floor. Dr.
Okay.
Fitri to ask question. Please, very short.
Yes. Thank you, Prof. Wiku. I'm Fitri. I want to ask regarding the One Health implementation approach, how important is community participation, such as direct community empowerment in combating zoonotic diseases? For example, leptospirosis in Bantul. Is a program like an environmental cleanliness movement possible to control disease vectors? Is that important, Professor, considering that the disease can affect both humans and pets? In my opinion, programs that target the community and are given appreciation or prizes will make them more proactive. Thank you, Prof.
Thank you. Well, I'm sure that the answer would be important. However, we need to make sure that the knowledge are shared with the community, wherever the community is, not necessarily only in Bantu, not only about leptospirosis, but also for other important diseases. So make the language very easy to be understood by the layman, by the people, the regular people, not the biologists, not the professors, not the physicians. For regular people, probably uneducated, that's the challenge from the scientists, from the public, the humas in all local governments, to make sure that they can convey the message to the public, because at the end, it won't be able for all government officials at the local level to control everything in the community. It's better to empower the community by educate them, not necessarily in the school, but something with the digital, with the many systems of communicating channels. So that's the key. Thank you.
Okay, because time is very limited.
Yeah.
Thank you, Prof. Wiku.
Thank
give
you.
applause for Prof. Wiku.
Thank you very much, Prof.
Okay,
Ayan,
thank
and
you
all
once
audience.
again,
Thank
Prof.
you.
Wiku, for your
Makasih.
expertise. I would like to invite the last speaker for today, Dr. Farida. Bu Farida Dwi Handayani, SSC MSc, is researcher at the Ekman Institute Research Center for Molecular Biology, National Research and Innovation Agency, BRIN, Indonesia. Her research focused on microbiology, molecular epidemiology, and zoonotic diseases, particularly rodent-borne infection such as Leptospira, hantavirus, and rickettsiosis. She has extensive experience in molecular diagnostic of One Health-based research that explore the interconnection between human, animal, and environment. Through her collaborative project and scientific publication, she contributes to strengthen zoonotic disease surveillance and advancing molecular research capacity in Indonesia, supporting national and global effort to prevent and control emerging infectious disease. Time is yours for Bu Farida. Please come to the floor. Yeah.
Thank you very much, Prof. Wayan, for the introduction. Hello, everyone. Good afternoon. Good morning, maybe in Sweden, if there is a Prof in Sweden. Okay, now I would like to share to you about our finding. Actually, I'm Farida from BRIN. So as we know, there is a new institution, actually, Badan Riset dan Inovasi Nasional atau BRIN, or we call it in English as National Research and Innovation. I'm under Eijkman Research Center for my molecular biology. So thank you very much for inviting me, Prof, because this the theme is very me like very interesting. We call it as disease outbreaks in front of our door. It is truly it is true that many diseases in front of us. However, maybe we have to find it. Okay, today I would like to present about the leptospirosis and hantavirus infection transmitted by rodents. So I would like also to present based on our study. We call it Pestorita. Why Pestorita? Because this is detection of plague or pest, and then leptospirosis, rickettsiosis, and hantavirus. So it is acronym of these diseases. From this disease, from this study, actually, I already got the whole genome sequencing of Leptospira culture or isolates from patient and rodents also. However, I think in this afternoon, we just focus about the leptospirosis in rodents. Oh, I'm sorry. Gimana nih? Oh, bisa bantu? Yeah. So for the outline, I would like to introduce about the leptospirosis and hantavirus in a very brief, and also about the Pestorita study itself, and also about the aim of study, the summaries and finding, and of course for the future direction in terms of One Health. It is very interesting because leptospirosis and hantavirus also as zoonotic diseases. So for the introduction, plague or pest, leptospirosis, rickettsiosis. In this term, we try to find the murine types, and hantavirus are diseases in humans and animals, or we call it as zoonosis, that are concern and priority to the Ministry of Health in Indonesia. They are still largely unknown, and the Ministry of Health requires research to support for early detection of these diseases. Plague, as we know, is a quarantine disease and a potential bioterrorism agent. And also, I will highlight the leptospirosis. This is a deadly disease, often misdiagnosed and still underdiagnosed in our country. This is very similar with dengue fever and typhoid. So that's why kind of like misdiagnosis for these diseases. About the rickettsiosis, but I don't want to talk about this afternoon. And the hantavirus, I will talk about this, is currently a concern because it was only recently discovered in Indonesia and has the potential to be prevalent based on the previous research. So these 4 diseases, similar symptoms and are transmitted by rats. This is very interesting because we know rats are abundant here, yeah? And then, so that's why we call it as rodent-borne diseases. So we talk about the global challenges and epidemiology of leptospirosis and hantavirus. For leptospirosis, this is a bacterial zoonosis caused by spirochetes in the genus of Leptospira. It is also known as rat urine fever or whale disease if it is severe. And also the estimated cases is about 1 million cases and contributes 60,000 deaths annually. In Indonesia, we have CFR around 5 to 15%, which is pretty high. How about the hantavirus? Hantavirus caused by orthohantaviruses. This is our envelope negative strand RNA viruses belonging to the genus orthohantavirus. And then this is also zoonotic disease that transmitted by rodents. So it is interesting, in our result, we can find one rodent with these 2 diseases. We found the Leptospira interrogans and orthohantavirus. And however, the case fatality rate is not too high, only 1 to 2%. So if you see the what is that the name? The table here in the right, this is a resume of the Pastorita research in 2023. We have 3 location or sites, which is in Semarang, Demak, and Pasuruan. Semarang and Demak known as the endemic areas of leptospirosis. We found 20% in Semarang for leptospirosis, and we found also hantavirus 6%. And, it is interesting, in the almost 20%, so 19%. And very interesting that we found hantavirus is pretty high in 31%. For Pasuruan, so Pasuruan known as a focus plague area. For the focus plague area, we never heard or we never received any report for leptospirosis. However, based on this research, we found 9% of leptospirosis incidents, and for Hanta is 11.8%. So for murine types, we didn't do the PCR or antigen detection. However, we did the seroprevalence using the IgG anti-rickettsiosis TPE for murine types. Interestingly, we found percentage almost like 20% for each area. That means we have the disease over there. So this is the resume. However, I will explain later. However, from the map, we can see if the distribution of leptospirosis based on the Ministry of Health, only several provinces that reported the leptospirosis. We don't know, maybe the gray areas is underreported because we know the all area of Indonesia based on of study from Ricus Vectora from Ministry of Health, we found percentage of Leptospira pathogenic or interrogans from the rats. Okay, so what is the gap? Actually, for these 2 diseases in Indonesia, we have difficulties in early diagnosis, especially early diagnosis in healthcare facilities. And also, we need actually the rapid and early laboratory detection, and then common community behavior support from village, sub-district. We need their support, and then cross-sector commitment. So this is some papers that I really love to show you about the leptospirosis in Indonesia. We have challenging in diagnosis, and then only limited laboratory capacity that we have. This is from Professor Gasem, and also for plague and for hantavirus. This is some paper that very interesting that we can see about the murine typhus also in Indonesia. It is very interesting because we know based on other research there were percentage of murine typhus in patients. So about the rats, why rats? Because we know the rat's reproductive cycle is very quick, yeah. So the sexual maturity, so only 5 to 8 weeks of female rats can mature, and males around 10 weeks. And it is interesting that rats have estrus cycles about the 5 days, and then after after giving birth, they can immediately spontaneous ovulators or pregnant immediately after giving birth. And then also the period of pregnancy is very short, only 21 to 23 days. And the litters on maybe 6 to 13 young mice or pups, all even more. And then postpartum estrus, only like 2 days, even 24 hours, rats can mate, and how can I say, they can get pregnant again. And then so basically from, oh my God, yeah, basically from this picture, we can see from 2 rodents in the short time, it can be multiplied a lot. So that's why if we just, how can I say, we only try to control, not by holistic, it will be very difficult. Okay. Now about the rodent-borne diseases. So, rodent-borne disease, it is very interesting because rodents represent the most diverse and comprising 2,200 species, nearly 42% of global mammalian biodiversities. It is very interesting because the species is a lot, and there are major reservoirs of zoonotic pathogen, including viruses, bacteria, protozoa, and fungi, and are particularly effective to transmit the disease. So we call rat as reservoir. And also rodent-associated diseases that impact public health includes bacterial infection, for example, the leptospirosis itself, and also Francisella tularensis. This is very dangerous disease also. For the another bacteria, we can see the plague and bite fever. How about the viruses? Viruses, it is include the hantavirus and rickettsia, also the parasite. So how rodents transmit diseases? It can be direct contact by handling infected rodents or being bitten. It can be also transmitted by their urine or feces or saliva, touching the surface contaminated, touching the, how can I say, touching the surface contaminated with rodent secretions. Also inhalation for hantavirus, the particle from dried rodents or their feces also can be transmit the hantavirus, and also contaminated water or food by consuming it. So in rodents, they have ectoparasite. We know they have 5 ectoparasite: fleas, ticks, lice, and it can be as a vector. So in this disease, we also try to pull the fleas of the rodents in Pasuruan, in the foci area of plague, and then we did the PCR for plague potential disease. So this is very interesting, this paper. So they mention about the hidden threat about the rodent-borne viruses and their impact on public health. This is pretty new and fresh paper from MDBI from viruses. I think in May 2025. This one. This is a paper, very just published in May 2025, and they mention about approximately 15 and 205 rodent-associated viruses in the 32 viral families. So it is belongs to this, including Hantaviridae family. And also this is the cycle, how the hantavirus or viruses can be transmitted from rodents, environment, and human. And this paper also very interesting for me because in this, they mention about the epidemiology, management, and surveillance, the controlling the leptospirosis, how leptospirosis, how can transmit between dogs or animals to environment, to rats, and to human because of our activity close to their animals. So how about Pastorita itself? I would like to share some results or findings that we found from this research. Again, this is acronym from pest or plague, leptospirosis, rickettsiosis, and hantavirus. Why we did this research? Because as we know, more than 30 diseases carried by rats. So we try to figure out for these 4 diseases or zoonotic. Okay. And this is our collaborator. So I received this research from BRIN and also WHO. And also Amsterdam UMC, thank you very much, and Institut Pasteur who help us in detection for plague and leptospirosis. So about the plague, maybe a very brief introduction about the plague. This is a quarantine disease, and plague was endemic from 1911 to 1947. And in Indonesia, we have 3 areas actually who still pointed as foci of plague, which are Boyolali and Pasuruan, also Yogyakarta, actually Cangkringan. There have been no reported cases of plague in Indonesia for over 50 years. However, the WHO try to still encourage us to do the surveillance of this plague because we don't want to have a silent periods of plague transmission. So it can be like outbreak also. For leptospirosis, so leptospirosis occurs in both tropical and temperate climates, tend to be underreported. And also the estimation that I was explained before, it is about 1 million cases annually, and also the death is increasing up to 60,000. How about the rickettsiosis? So rickettsiosis and leptospirosis are predicted to be endemic in Indonesia because based on inner response study, cohort study showed that rickettsiosis are often misdiagnosed as leptospirosis, dengue fever, and Salmonella typhi infection. It is very interesting and important study in Indonesia. And about the hantavirus, so the epidemiologically, orthohantavirus infection and leptospirosis are similar, very similar for clinically. And then orthohantavirus infection are frequently reported in leptospirosis endemic area, which is true because we found in this study also. So the aim of the study actually to determine the seroprevalence, distribution, clinical characteristic, and etiology of individuals with acute fever. So the start of this study is based on acute fever patient suspected of having zoonotic diseases. We don't know yet which is this is lepto or hantavirus or others. So this study we did both from human and animals. The second aim was to obtain cultures of pathogenic Leptospira serovar circulating in humans and animal reservoir, and characterize them by genomic with the whole genome sequencing. And luckily, this year we have the whole genome sequencing data already that we would like to analyze further. So the third aim was to determine the characteristic of the sylvatic environment. Sylvatic environment is the far or the far area from the household, including we would like to determine the host factor agents, abiotic and biotic factors, including mapping of the plague in Pasuruan Regency, East Java. However, I think we don't want to talk a lot about this third aim. So this is basically the location of our site, our study in Central Java. We did in 2 regencies in Kabupaten Demak and Kota Semarang. So both of these locations are endemic of leptospirosis. And if you see the map below, which are we found the rats with the red dots, and also the patient and the puskesmas or the public health center. So we try to overlay where we did the capture of the rats with the collection of the patient. And this is the location in East Java, is in Pasuruan. So Pasuruan, again, this is the foci of plague in Indonesia. So we are trying to catch the rats, collect the patient bloods to evaluate the leptospirosis also and hantavirus, also murine typhus, as well as plague. So this is the finding of research of leptospirosis in rats, because I would like to focus only in the rats. So for rats, we did only the conventional PCR. However, for the patient, we did the real-time PCR, because in the rats, we collected the kidneys to get the DNA. So that's why endpoint PCR or conventional PCR is enough to detect the leptospira. So in this slide, I'm sorry. So in this slide, we can see several rodents positive of Leptospira interrogans, which is the agent of leptospirosis. And we found in the habitats of whole farm and forest. So it is interesting. We can see the, how can I say, the positivity rates. And also for hantavirus, here in Semarang, I saw you in Semarang data that the prevalence of captured rat and trap success in Semarang is pretty high because we found in Semarang, in Kelurahan Kecamatan Gayamsari, we found two rats, which is Ratus norvegicus, tikus got, ya kalau bahasa Indonesia, and Ratus tanezumi is household rats. And then also we found Suncus murinus. Suncus murinus is not the type of rats, but insectivore. And I don't think so the Suncus murinus as a reservoir of leptospirosis or hantavirus. In Telogosari also, we found Ratus norvegicus or tikus got and Ratus tanezumi. So this is the data from capture rat. Ratus tanezumi and Ratus norvegicus is the most abundant in Semarang. How about the prevalence of orthohantavirus? So this is the findings of hantavirus by PCR. So we found Ratus norvegicus in Gayamsari. We found 1 out of 9 positive for hantavirus from their lungs. However, we didn't see or we didn't find in Ratus tanezumi because this is the cross-sectional research. Maybe we can do the further research for this. And also in Telogosari, we found in Norvegicus, Ratus norvegicus. So the incidence of orthohantavirus in Ratus norvegicus in Gayamsari was 4.7%, and overall incident was 2.38%. Hantavirus target gene is the L segment, which is in 388 pair. And this is the picture of our results. And I think to wrap up the result, this is our future direction about the One Health in leptospirosis and hantavirus. So a One Health approach to the epidemiology, management, surveillance, and control leptospirosis relies on accessible and accurate diagnostic that can be applied to human, animal, and livestock, of course. And then this One Health approach we thought should be implemented in the leptospirosis and hantavirus control and should be implemented nationally by harmonizing the program across sectors that Prof. Wiku just presented. It's very interesting presentation from Prof. Wiku, and who has responsibility for rats control. This is our job together. So anyway, it is very interesting that I have been invited by Vikia UNER Banyuwangi to give lecture in co-assistancing for veterinary student and try to find Leptospira interrogans from the cattle and rat kidneys. It works well. So we can find the Leptospira both in kidney of cattle and rat. So can it be a way for animal surveillance? It is very interesting. So this is our pictures with the students, veterinary students from Vikia UNER, calon-calon dokter hewan ya. Jadi stase koasnya ada di situ, dan they learn how to detect leptospirosis, how to do the PCR. So I think that's all that I can share this afternoon. Thank you very much for One Health Collaborating Center UGM, Prof. Wayan and team. Thank you for my institution, of course, and Ministry of Health and our colleagues abroad. And this is our colleagues in Ekman Research Center for Molecular Biology, and this is our colleagues from Amsterdam UMC, from ICM Amsterdam, India, and also nationally. Thank you very much for your attention.
Okay, thank you, Bu Farida,
Thank
for
you,
your
Prof.
presentation. Very nice presentation. But today we do have outbreak also on leptospirosis in Yogyakarta.
Today,
So,
Prof?
yeah,
Recently.
200 cases in Yogyakarta of leptospirosis. So regarding rat as a reservoir of the leptospira, there are many species of rat. But what, as you mentioned, that Ratus norvegicus and Ratus tanisomi, one of the reservoir, the best reservoir for the leptospira. How about the other species of rat like Bandekota, for example, and Celurut? They also they also transmitted the leptospira to the human being.
Okay. Yeah. Thank you very much for your question, Prof. Wayan. Actually, you already know the answer, but yes, thank you. I would like to explain a little bit more based on our study. Previously, when I was a researcher in Ministry of Health, we had a very huge studies about the leptospira in rats. And interestingly, Prof, we found many species of rats can transmit the Leptospira interrogans, not only Ratus norvegicus or Ratus tanisumi. Bandicota indica, as you said, is Ticus wayrock, the big one. Is also we found the percentage of Leptospira interrogans in the kidney. So it means it can be delivered, it can be spread to the environment, and of course people can get the infection of this disease. And also tikus sawah, or we call it as Ratus argentiventer, also Prof, we found a lot of pathogenic leptospira in the kidney. However, why we didn't find in this study? Because we only do in the household, not as Ministry of Health Ricus Vectora. We did from the coastal area, household, and also the forest. So we can catch the all capture rats from this area, not all, I mean like the percentage of the population. And then we surprisingly we found also forest rats, household rats, and coastal rats also, Prof. We can find the Leptospira in the kidneys because they are reservoir. Basically, the all species can be a reservoir. However, as you mentioned about the Suncus murinus or cecurut, we call it in bahasa. Cecurut can be cannot be as a reservoir of leptospirosis. Why? Although some study mentioned about the positivity in kidneys of Suncus murinus, however, their urine has acidic pH. So that's why the leptospira will be killed through the urination process. So we don't include the Suncus murinus as the reservoir. Also, percentage of positivity we found in their kidneys. Thank you, Prof.
Okay. My second question, because no one asking the question, my second question regarding the seropar. According to our study in Bali, we found that in the human being, there are triple infection, Leptospira canicola, Leptospira erythrohemorrhagica, dan pulmona in one patient, in one
patient.
human. How come this? Because the erythrohemorrhagica come from the Ratus norvegicus, and then canicola from the dog, and pulmona from human being.
Okay.
How come that three possibility? Because we did found out three species of Leptospira.
Yeah.
Seropar, three seropar.
Thank you, Prof. It is very interesting. If we talk about the microscopic agglutination test, which is a gold standard of leptospirosis, actually we talk about the titer. It is interesting for me to compare these three titers of the infection. I believe based on the clinician explanation from Prof. Hussein Gassam, let's say, uh, Prof. Gassam mentioned only one serovar can infect in one time. However, why we found two other serovars? Because maybe the background, Prof. So that's why actually the current infection based on the MAT should be fourfold rise titers. So we are maybe like more than 1 in 300 to 20 in titers. So they will have one, the highest titers, which is the current infection, and two other serovars, maybe the background. So this patient may be previously infected by another serovars, because usually we can still see the titer of the past infection during our MAT. So that's why, Prof, sometimes this is the interpretation of MAT when we only have one sample of acute sample blood or sera, only one, but the titer is not reached up to the 320 and we said it is negative. No, we cannot say like this. Actually, we need two pair sera, which is acute and convalescence one. So that's why MAT for me, myself, it's very difficult to do in Indonesia. So I think recently it will be very easy to see the serovars by whole genome sequencing. Prof. Wayan is very expert in this field also, and maybe I will learn from you. Thank you.
Okay, thank you, Bu Farida. Any other question from the floor here? Bu Sabrina, you want to ask question? The student? Bayu? Yeah.
Okay. Hi, Ms. Farida. I have a question about the diagnostic. As you said earlier that currently we still have a difficulty in establishing the diagnostic for leptospirosis. So my question, what makes it difficult to early detection of leptospirosis, and what is the current diagnosis test for leptospirosis? If I'm not mistaken, you mentioned about the real-time PCR, perhaps, because as long as I know, that tool is very expensive, and perhaps for some people it is not affordable, and also time consuming perhaps. So yeah, that's my question. Thank you.
Thank you very much for this very important question, actually. So about the two how can I say, two questions about the leptospirosis diagnostic, about why difficult to diagnose in early infection, because leptospira is very thin, very small, and very quick to move to targeted organ. So I found the differences between the serovar maybe in Semarang or Demak and Balaraja in West Java. So in Demak, let's say, the leptospira is very quick to move from the blood to the targeted organ, let's say kidney of patients. So that's why before the antibody titers is rising, the leptospira itself cannot be detected by PCR in the blood. I mean, like difficult, not cannot be, but difficult to find in blood of the patient, because they goes to the kidney already. However, the titer of antibody still very low. So the RDT, or the rapid diagnostic test of leptospirosis that available only based on the IgM, it cannot be detected. It can't detect yet. You know what I mean? Like, so, and then for the, how can I say, for the Balaraja, it is interesting because the leptospira stay longer in the blood. So, so that's why maybe it is challenge for leptospira diagnosis. And then about the current detection you mentioned, yes, PCR. I think is the best
Kali ada ragil.
way to do right now because the RDT, IgM based RDT cannot rely on the early detection in 5 days below. So that's why we have to do the PCR. So we have serology by RDT, IgM based, and we have M80 for the gold standard. However, we need the two samples, acute and convalescence, to confirm. And then for the molecular technique, we can do the PCR, both conventional or real time. And also recently, I tried to develop the antigenic rapid test together with Conimex actually and US. And then, however, we already have the kit in the lab. We try to detect the leptospira. It can work well. We can detect it up to 10 to 4-fold for the leptospira. However, in the patient, it is not sensitive yet. So this is our challenge. Thank you for question.
Bu Farida, here are another 2 questions from the chat box.
Okay.
One question from Dian Puspitasari. Thank you for the interesting presentation, Bu Farida. My name is Dian. I come from human medicine background. What are the implications, perhaps practical ones, of your finding on leptospirosis research for the people in the medical profession that deal with human health? Thank you. Should I again? What are the implications, perhaps practical one, of your finding on leptospira research for the people in the medical profession that deal with human health?
Maaf Prof, nggak terlalu dengar karena ini boleh saya baca langsung nggak Prof? Boleh ditampilkan panitia untuk ininya? Yang mana ya?
From Dian Puspitasari.
Ya, kasih. Maksudnya dalam bahasa boleh nggak Prof? Saya nanti
Ya, boleh.
Gimana Prof? Saya
Ya.
juga. Bahasa Indonesianya apa Prof pertanyaannya?
Ini Dian.
I'm sorry because I don't have my Zoom link here, so maybe.
Oke, jadi pertanyaannya adalah apa implikasi mungkin dari sisi praktikalnya penemuan-penemuan ibu terhadap leptospirosis, terutama mereka yang bekerja sebagai praktisi di bidang medical seperti itu.
Oh yeah, so what? Okay, I'm working with Prof right now for my PhD study in biomedical sciences in Faculty of Medicine Universitas Indonesia. I'm working with Prof Erni Cwitanelwan and also in Semarang with Prof Hussein Gasem about this leptospirosis. They mentioned about this importance of knowing the infection in rodents, also in animals, because it makes them think about the transmission around the patient household. So maybe it can be as a clue for the clinician about the existence of leptospirosis or hantavirus itself in this their area. So I think this can be as a early warning system, maybe, Prof, that we can get from findings from the animal to the human. That's I think it is my answer about this, and also based on my finding for my dissertation. Also, it is very interesting that we found difference typical of infection of leptospirosis between Central Java and West Java. Whether in Central Java, we found based on the clinicians, the patient is very quick to be a severe leptospirosis. So only 3 days of fever, they can get into the ICU. However, in Balaraja, Prof. Ernie Juitanel one cites, the leptospirosis cases is mild, Prof. So we still can find leptospira in the blood, although 10 days, 7 days, which is it is impossible we found in Demak or Semarang. So maybe I thought it is we have different pattern of the whole genome sequencing, or maybe the difference of serovar that infect the patient. So that's why, based on our result, we would like to do the further analysis on serovars. I hope I answered the question of Ibu Dian. Thank you very much for your question. Yes.
Okay, this is the last question. I will read it in bahasa. Pada sapi itu juga terdapat potensi yang besar karena sapi berhubungan langsung dengan tikus seperti itu. Namun pada kenyataan di lapangan, clinical case-nya itu relatif jarang dilaporkan. Menurut Bapak Robi itu diasumsikan karena pada sapi itu bersifat subklinis, gejalanya subklinis. Walaupun itu dapat menyebabkan penyakit, menyebabkan dampak pada reproduksi seperti aborsi, kemudian fertility, dan lain sebagainya. Pertanyaannya adalah, apakah angka yang rendah pada sapi benar-benar menggambarkan low prevalence-nya, atau hanya karena imun si sapinya itu yang dapat mensupres clinical manifestation?
Thank you for the question. It is very interesting, and I did prove by our study also, Prof. So yes, true. In human and dogs, we are the end host. So we can be killed or we can die because of this infection. However, for other animals like cattle or sapi, especially rodents, the clinical symptoms is only subclinically symptoms, because they didn't show any clinical the true or the tampak gitu ya, tidak tampak. However, in kuda or horse, they can be blind. They can blind, or we call it as, I forgot the term, Prof, for blindness in horse. Uveitis. This is horse. However, for the cattle and kambing, it can be abortion. It can be abortion. So that's why it is very difficult for veterinarian to think about leptospirosis. But our collaboration with Vikia Uner Banyuwangi, we found that in the kidneys of the cattle, we found percentage of leptospirosis, and the students got the kidneys from the slaughter, ya Prof. RPH itu slaughterhouse. So I'm thinking if we can adopt this to be it can be a surveillance system maybe in the cattle. So maybe if in the area with the positivity of the leptospirosis in the cattle, we can do the surveillance. we can also do in the urine of the kettles, actually. Thank you, Prof.
Okay, thank you, Bu Parida, for your nice presentation and also sharing your experience with us.
Okay, thank you very
Big
much.
applause for her. Okay, thank you everyone to join our webinar for today. I give the MC back to MC.
Thank you very much, Prof.
Okay, thank you, Miss Farida. So, ladies and gentlemen, we have now reached the end of our webinar and discussion. We would like to express our deepest appreciation for all speakers and Prof. Wayan as a moderator and also all of the participants across the world for your active participation in this webinar and discussion. We hope this event has enriched our understanding of the links between climate change, eco health, and also zoonotic diseases risk. With that, we officially close today's webinar and discussion forum. Thank you so much and have a great day. See you on the next event. Thank you.
Share the air, we drink from the stream.